where separations has been in the laboratory and pharmaceutical equipment supply and sports space for the last 3 decades. Jeff is a biochemist and joined separations in 2010, working in the pharmaceutical support division, having worked with majority of pharmaceutical clients in the country and into Africa. So Jeff, welcome on board, and yeah, good to have you.
Thank you so much for having me. I just want to confirm that I have my screen shared,
Yes.
that everyone can see me. I'm just switching over. Perfect. So I should be in presentation mode. Excellent. Good. So obviously, thank you for the introduction and thank you for the invite. I'm going to discuss an overview on the cannabis and hemp extraction platforms that are currently available. And then for me, it's really important because ultimately, why does extraction matter? When we get into this space, especially currently in the medical markets, you know, where you ultimately have an offtake for your product, it's usually going to go off either as a distillate or potentially an isolate. And if you don't understand the extraction process, you're going to end up in a situation where there might be pitfalls. And considering, you know, there's been a lot of questions and need for clarity around legislation, the last thing you want to have as an investor or anyone in the business is not to be sure about what do we do next. If we cultivate, there's a lot of good cultivation experience in the country. What happens at the next stage where we need to actually extract and supply? And very often I get 1 or 2 questions from stakeholders, which is, do I just take the flower and I put it in a machine and on the other end I get edible gummies, which is not the case. And then secondary to that is a lot of stakeholders would say, well, I'm not too sure, is it super complicated? And how do I ultimately get an extract to work? So today, what I'm going to try to do is, in the space of half an hour, which is tight, give you a real breakdown of what you need to know to be able to facilitate extraction and the relative frameworks. So I think with that, let's get started. I'm going to quickly touch on cannabis extracts. What are they? What do you need to consider when we do an overview from flower to finish, being the final dosage form? Pre-processing considerations, how do I get my cannabis ready for processing? What is the best extraction platform, which is an important question? And what are post-processing steps that we need to take and possible Q&A? So just some background, as separations, for anyone who doesn't know us locally, we've been around for quite a while and we have a team of about 70 scientists, salespeople, engineers, application support specialists, and we have multiple divisions. Myself, I'm in the pharmaceutical side of the business, but for instance, our diagnostic division has been very involved in the COVID response and automating the testing for the extraction robots to be able to increase those test volumes per day. So we tend to take a very long approach to projects. So we are targeting to be in this space for the next decade, and as such, we've partnered with world leaders in extraction, such as Apex Supercritical, who's got 600 systems in the market globally, and Delta Separations, who's got 800 systems in the market globally, and working with their teams and their experts to ultimately provide the best extraction and post-processing platforms to clients that will meet international standards. And especially in the medical space, meeting international standards is paramount. So I get this question daily from stakeholders, which is, which extraction platform is the best? And it really comes down to the type of product you're going to supply. So if we are talking about a distillate or an isolate going off to an international offtake agreement that needs to conform to EU GMP, there's a completely different approach. And hopefully in the future, we could also see the expansion around the nutraceutical extraction space, which is probably likely next, and then possibly even in the future, recreational space. So to understand which extraction platform is the best, we need to know a bit about extracts and also a bit about their dosage forms. So this is typically seen where you have cannabis concentrates in the market, and the main ones we would focus on, and I'd like to just point to this, just want to see if I can get into points of view. Let me pull up the laser. Typically distillates and isolates would be the main discussion in medical, and possibly terpenes due to the entourage effect. But in the more recreational space, we'll see things like crumble, shatter, sieve, rosin, bubble hash, fresh frozen. You need to be aware of these distinctions because the extraction method you choose ultimately gears you towards a certain platform. So we'll focus more on the distillates and the isolates discussion and workflow. When it comes to the final products, At this point, we are really focused more on the medical supply and support space, so distillate and isolate, and possibly encapsulation going to soft gel or tinctures. But the big future push will no doubt be in the consumer space around food, consumables in terms of beverages, edibles. This will be a big spin in the future of the market as CBD as well as other minor cannabinoids work into the legislative framework. And then the recreational market obviously opens several opportunities, as is well realized in Canada and the USA. Now, this will be the most dense slide I will show, but I think it's still important because we'll have a bunch of individuals that are very unfamiliar with the extraction workflow, and there will be others that have been involved in the space for a while. And ultimately, what we look to discuss is there are really 4 primary considerations in extraction. And it comes to your pre-processing, which is basically how do we prepare our cannabis for extraction? Then we come to the extraction platform. Once we've prepared our cannabis, which platform do we proceed with? And then finally, depending on your extraction platform, you also will have post-processing considerations. So certain extraction platforms will pull in a lot of lipids and waxes and fats, and you might not want that in the extract. You might want a very clean distillate. And if that is the case, there are certain post-processing steps to be applied. In this space, and very popular in the US, is a concept like THC remediation, where there you would need to then process that further to make sure that if it was a hemp-derived product, that it can be processed, and if it is allowed for, legislation would ultimately allow for that post-processing. And then finally, all of this only comes into effect when you have a white label filling line, or if you're doing white label for a contract manufacturer and understanding your packaging. So the big highlights here I would like to just touch on are, how do I mill and grind? Is decarboxylation important? Are we looking at a mechanical extraction, which involves different platforms? Are we looking at a supercritical extraction, or an ethanol or butane or solvent-based extraction? And then all these topics that can possibly apply to the post-processing will relate to winterization, filtration, possible chromatography steps, definite distillation steps. And from that slide deck here in front, you can see what's on that topic list. And I'm always happy to engage with this in more detail. Right, so the biggest concern to any cannabis cultivator is you're going to go and you're actually going to mill and grind my flower. And this is usually counterintuitive in the space because those beautiful flowers and buds tend to be, it's what you grow and ultimately aim towards. So milling it is a bit of a counterintuitive feel, but when it comes to processing, if you're not able to sell certain flower formats in terms of an international offtake, you still have additional material that can be processed in terms of bioprocessing. So entering into the agro-processing space. The important consideration here is when we mill grind material, we want to get as even and as consistent a grind on that material. The reason being that whenever we apply this to an extraction, we want to almost deal with the same material every time. We already know that biomass is going to be very variable. You might have one cultivar where you put in a certain amount of THC to CBD or vice versa, and that already creates a bit of a problem in terms of managing expectations for production. So the one thing we want to do is we want to control the extraction process as closely as possible so that we can limit the effect the extraction process has on the quality outcome, and ultimately then we're more reliant on the actual initial biomass. It is a science and it is a bit of an art, but the first steps you can take, and it's very often overlooked, is making sure that your material is as homogeneous and consistent as possible. Where this is a good example of a pharmaceutical mold, and this would be maybe a more commercial mold, where you would grind your material to the consistency you need so you can then process it. The question that often comes up is whether or not to decarboxylate the material, and often decarboxylation is the chemical reaction which involves the removal of a carboxyl group from the cannabinoid. This often applies more to THCA and CBDA. The main reason it's important is if I decide to decarboxylate THCA, I end up with THC, which is psychoactive, whereas THCA could have medicinal purposes and not be psychoactive and quite attractive in terms of final format. So it really also has to play with the terpenes, where the entourage effect comes into play with terpenes. And if you're decarbing your material and you're losing those terpenes and not capturing it with a cold trap or taking a unique approach to it, you lose value. So very often with the supercritical extraction system, what we'll do is we may decarboxylate the material, strip the terpenes in a subcritical run. And if this all sounds Greek to you, I will try to make this as clear as possible in the next slides. And then from that, we have our terpene fraction, which will facilitate the entourage effect. And then we can then take that and decarb the material once we strip it from the extractor, and then focus on extracting our cannabinoids in the form we want. And then when should I decarb? This is relevant for supercritical extractions, usually ahead of time. And then when it comes to ethanol, we usually would do it in a reactor further down in the process. This recording is being recorded, so if I am going through material quite quickly, I'm trying to stay to the time limit, but you'll have access to it. So which extraction platform should I go with? Well, what are we trying to do? We're trying to target in on specific cannabinoids or specific terpenes. When it comes to, I really just want a cannabinoid-rich distillate, and that's really what I want, and I want to possibly take that distillate to isolate. Ethanol can be a good considerator there. I would say if we're looking for a more full representation of the plant, and we want the terpenes and we want additional flavonoids that is pulled along with the cannabinoids, then the use of something like a hydrocarbon system or a supercritical system really facilitates that more efficiently than ethanol. So that's something to consider. The other point to consider is concentration. Whenever we are extracting, we usually are looking to extract and concentrate a sample. And with that, the source biomass or material also concentrates. So we concentrate potentially things that we don't want. So the quality of your biomass is really important in this consideration, because you don't want to be concentrating things like pesticides or heavy metals in your extraction. And that's why we also offer testing solutions, but it's just to make sure that we have high quality biomass. Now, for those out there that have heard anything about extraction, we usually have mechanical and solvent-based extraction, and there are usually proponents in each camp on which is the better or the preferred method. As I said, there really isn't a better or preferred method. It really comes down to what you're trying to achieve. For mechanical separation, we usually target the removal of the trichomes from the actual bud. So things like a dry sieve, where we use a mechanical sieve and we break off trichomes, is very similar to concepts like ice water hash, bubble bags, where we're just agitating and trying to remove the actual trichomes, which contain a lot of the terpenes and the flavonoids and the cannabinoids. And then things like a vortex trichome separator uses bioagitation with water to do a bit of a water-based solvent extraction. And then there's things like rosin press, where we apply heat and pressure to extract our oils. And then things like screw press gets found in more industrial applications, where we might be processing and squeezing hemp in that regard. The more commercially viable extraction platforms tend to be solvent-based, and the reason for that is the solvent really gets to get in there. So a solvent is able, if you prepared your material correctly, a solvent is able to get into contact with your product or compound of interest and really strip it from that material. And this is really particularly useful in terms of CO2, where we could have subcritical and supercritical runs. We could use it in ethanol, where it's usually run cold, and I'll explain the reasons for that, where we strip also from that. There's hydrocarbon, there's larger pentane extractions, hexane extractions that we considered. A lot of home users might have tried MCT oil. It's not an ideal carrier necessarily for extraction. It works, but there are far more viable commercial extraction platforms like the ones discussed on top. And naphtha and other refrigerant gases, there are always new solvents being tested in this space, and even interesting concepts like centrifugal partitioning chromatography, where what we're doing is we're basically using a chromatography system that's in a centrifuge, and what it does is it allows for a mobile and stationary phase to allow for good selectivity. So using a system like this, you could potentially target specific cannabinoids in the initial extraction, which is quite useful to be able to do. It's just a question about scalability and costs. So the more commercial options are our CO2, our ethanol, and hydrocarbon. Right, here's just some images I wanted to show for those who aren't familiar with what I've mentioned, which is a rosin press. An industrial screw press would be very large and would work on something like a screw that just twists and squeezes the material. Bubble hashing, ice that's often used and introduced into this, and these are usually methods that are mechanical or solventless. So when we come to solvent-based extraction, which is more commercial, this is just an example of what a supercritical CO2 extractor would look like. And to break it down, I'm just going to show you what would ultimately happen in a system like this. Is CO2 would be pulled from the system, run through a compressor, and the program would dictate this. And what it will do is it will push CO2 either subcritically or supercritically over your biomass, which you would load into these extractors. Now, this is a relatively small system, and they get quite large, the type of CO2 systems that are available. But the most important thing is what happens in your actual separators. Because in your separators, you allow for the specific precipitation out of certain fractions. So the nice thing about supercritical is you allow for fractional extraction, where often other solvents you just extract with that solvent, and then you have to do a lot of post-processing to get the fraction or the product of interest. In supercritical, because you're manipulating temperature, time, and pressure, you can often fractionally extract and allow for certain compounds to be precipitated out into your cup or your receivable collection flask. So this is typically how it would work, and you would recycle this as a closed loop system and reuse your CO2. And there's a chiller and heater to drive the parameters. And I want to quickly touch on pros and cons of the different platforms, because I want to be as balanced in comment on this as possible. The pros initially of supercritical is that you get good selectivity and good flexibility. So like I mentioned, you could do a terpene or subcritical run where you strip this. You could do a nug run as well, where you actually load buds as buds, but ideally to get better efficiency, you might decide to mill that material. In addition, you could have a run that's specifically putting more waxes and lipids along with cannabinoids, and maybe something that dials in a little more with this. And this is why we work with international collaborators, so that we can really set the SOPs and dial in the parameters so we can get the best extraction parameters and results. The nice thing is with crude, you often don't have any residual solvents present, which is a bonus. However, if you are taking this to winterization with ethanol, that is something to be factored in, and I'll discuss that. These systems are easily automated. It's very safe because CO2 is used to put out fires. It's not a situation where we could have a combustible material. So from a facility design side, these are pretty easy to implement into a more traditional clean room or appropriate environments. It has a good perception as a clean solvent, and this particularly applies to tincture bottles where you might have seen it says subcritically or supercritically extracted. You don't see bottles where they say it's been butane extracted. So this has more to do with marketing impact, but when we look at a business, ultimately we need to consider the market in that. So, if this is something that is applicable to your offtake or your final dosage form of white labeling, you may want to make this claim around the supercritical extraction. From a long-term perspective, the OPEX or the operational costs of supercritical is quite attractive. But that also plays against the negative in cons, where the biggest con and inhibitory factor about supercritical, and it's why it's normally dismissed immediately, is because of its high equipment layout costs. Now, I always try to say there needs to be balance in the fact that you also need to consider the facility costs. I'll go for that with the other cons, but that's a definite reason why it's not attractive. It costs a lot of money because you have to have vessels that are well engineered, meet ASSME standards. It's one where ultimately it's just well engineered systems and safe systems. They have lower extraction times relative to other platforms, and this is due to the selectivity I mentioned. Because you're targeting specific extractions, it could affect the extraction time overall. And scaling is difficult as well, because the larger you go, it's not like you could buy a small supercritical system and just scale it into a bigger one. You almost need to decide what is going to be a good fit from the onset. I'm not going to go into this because I don't think it's appropriate for the audience and the time we have, but this just shows, when is a supercritical, CO2 usually is a liquid gas or a solid. It can also be in a supercritical state, and this is just some of the science behind that. Where basically you have it as a solvent that's it operates like a gas but extracts like a solvent. So now butane, this is more familiar perhaps to the markets, and what are the pros? Obviously low equipment costs. Operates at lower pressures, which makes it attractive. It's flexible, so mixes of butane and propane can be used. It gives good yields. It has medium extraction times, and it's great for dabbing products. Some of the best live resin and shadow products would be from a butane extraction. The only thing is this tends to lend itself to the recreational market, and until that really is in full steam or allowed for, It's one where this is really not envisioned to be the immediate market. The cons, and this is specific to more the facility, is it's expensive to automate due to the explosivity related to hydrocarbons. The facility costs can be high. Very often you would want to house this in what is called the Class 1, Division 1 room or facility, so C1D1 room. And it's because of these safety considerations that it carries that hidden cost, for lack of a better word. So things like explosion-proof light fittings, HVAC that's really stripping the room of any exposed, or even if it's a closed-loop system, you need to make sure that you protect the operators. And the last thing you want to have, and any regulator would want to have, is an explosion on site at a licensed facility that ultimately puts back the industry from a perceptual point. So these are key considerations to be brought in. Something that is important is whenever doing hydrocarbons, the residual solvents and limits in that extract is important due to possible carcinogenic effects of hydrocarbons. So that's closely monitored, and that's where we also would test for. And the need for avoiding waxes and chlorophylls in the extraction drives lower temperature processing. Generally, there is a bad perception around this due to, let's say, poorly run operations that create a bit of a negative view on the use of hydrocarbons. And scaling can be difficult due to, obviously, the choice you make in the system and also the facility, how it was built and designed and configured. Right. This is just an example of, what we would typically look at in terms of room layouts and the rest of how the workflow will be for that facility. Ethanol. So this is our last primary extraction platform to consider. The pros is there is low to medium equipment costs. I would say it's more medium for the medical space than low. It does provide for high yields, which is great, and it also makes it attractive in the hemp industry space. It has some of the fastest extraction times. It's very commonly used and it's well accepted in other industries. So, the use of ethanol is well established and regulated, and it's relatively easy to scale. Anyone who's worked with hydrocarbons will know it's not that easy always to get hold of the gases, depending on the form of the gas. But this is relatively easy to implement and scale. And here's just a good example of a warm ethanol extraction where you put in a lot of chlorophylls, and then when you run your extractions a lot colder into the negative 40, negative 50 or colder range, you start to get a much better extract to work with that avoids post-processing. But it does have the cost of running a chiller to bring you down to those temperatures. The big cons here is moderate facility cost, because you still would want to operate ethanol in what would be class 1 Division 2 room or appropriate HVAC room. Chlorophyll also drives the need for colder processing. There could be some post-processing with filtration. Residual solvent limits are still applicable, and ethanol, depending on its taxation and its losses within the material, does have an impact on the commercial viability. But I'll give you a breakdown slide of that next. And significant post-processing energy expenditure does apply because you're recovering and evaporating that ethanol and bringing it back into the extraction. So this is just a quick breakdown for anyone who's looking at it from an investment point, from a costing point. Which extraction platform do I choose from? I'm always here to help facilitate that conversation. But generally, CO2, doesn't matter from which vendor, will have a high equipment cost up front. The facility cost will be low because it will basically be the minimal requirements of the free. Operating is pretty low because of the automation of the systems. And because it's closed loop, you just set the button, you go. The nice thing is, depending on the size of your extractors, supercritical systems could also be set to run overnight, and you just come in the morning and connect the fraction, whereas the other systems will have to be more closely manned. And then long-term solvent cost is the lowest on CO2. So that has a commercial impact, especially if you consider if you're losing solvent. That cost adds up as you commercialize the project. Butane, you can see medium, high facility due to C1, D1, operating costs, training, safety protocols, checking certain things within the environment. You have to pay constant attention to what you're doing with butane. Medium solvent costs, ethanol, low to medium across the board for the other considerations, because you still have a facility that needs to have an adequate HVAC and consideration for class 1, division 2. So, and this is based on roughly working in that more commercial scale per day of about 60 to 120 kilograms. And these systems also require the good use of pop chillers, because you do want to be efficient in your extraction. Right, and I won't touch on this because we're running a little tight on time, but essentially centrifugal partitioning chromatography is some of the newer technology that is coming out, but at a $2 million cost point for what is a relatively small extraction system based on what it can do. It is a bit inhibitory until the science develops. So post-processing, I'm going to basically touch on this where, there's quite a few things to consider. Based on your extraction platform, you need to know, how am I going to treat that in the post-processing? And anyone familiar in the space will know concepts like winterization exists, where you freeze and precipitate, and you remove lipids, where that can be scaled so that you can really get a clean oil. When you are working with ethanol in the winterization process, you need to remove that solvent. So things like short path distillation needs to be applied to really enrich and purify a higher purity of your cannabinoids, or it could be commercially done through a work film or spinning band. And then automation always exists in the space to produce a high quality product. To be respectful of time, I'm going to move through the next slides quickly because I really would like to engage on the questions and answers section. So for me, winterization is basically just that concept of removing your lipids and fats so you can get a high quality oil. And it's not a standard freezer, it's one where it's really a cold freezer, and we'll take you through that. There's a lot that goes into that equipment, and I'm happy to discuss that. There's the use of possibly activated carbon or other media agents that help you with the filtration and the cleanup and the coloration of your extract. This can be scaled for GMP, where we really move to lenticulars or filter presses. For the winterization, there's a lot that goes into it. One thing I would point out from a medical perspective is, even if you're stripping the ethanol and reclaiming it, you need to consider possibly how often you can reuse it, proving that the quality of that ethanol is what you desired at the start of the extraction. And you might have to also put batch and lot limitations on that, so that if you're doing toll processing under a licensed format, that you're able to do that on behalf of others and not have interference between batches. These are larger automated rotovacs we supply from Hydrocarbon, so this is a really nice system, especially when you automate it. So the nice thing here is you could have 300 liters arrive in a vessel. You leave it unattended, the system does it, and what happens is you come back the next day and you've got your distillate oil in one container and you've got your reclaimed ethanol in the next. This is really ideal from a lot and a batching perspective, and really a very good model if you're looking to automate your extraction, and more applicable to some of the future applications. Decarboxylation, which is the conversion I mentioned earlier, can be done either in rotovac or in a decarb reactor. And then we can go into things like short path and fractional distillation, but the thing you need to really know here is what we're doing is we're enriching what would typically be a 50 to 65% cannabinoid containing initial distillate to what would typically be a 95% or superior type of distillate. So, and there we have things like running really deep vacuums, hot and cold condensing technology, vapor sensors, and there's quite a lot that goes into this, but I'm happy to have this as an offline discussion, or possibly come back to discuss it in more detail. This is how it looks automated. I'm not going to get into topics like, the possibility that exists out there of converting CBD to THC, or even the chemical conversions that could exist about converting something like CBD into other minor cannabinoids. I'm always happy to entertain it, and I enjoy those conversations. For me, I would really like to get us into a question and answer platform quickly before the break. But I'm always ready to engage. So if you'd like to reach out to me, I'm always available on the phone. Even a WhatsApp works, so that I can initiate the conversation. And you can get hold of me on my email or head to our website. So these are my particulars. And I'm always looking forward to a good engagement and a good discussion. So I think at this point, what I'll do is, Dave, would you like to jump into some of the questions? I'm going to exit presentation mode on my end. Sure.
Look, Jeff, perfect. Don't worry too much about the break. I mean, we just scheduled for a 10-minute break, and we seem to be going at a good pace, so we don't necessarily have to have that break. We can probably have a 5-minute break if people want to just pop to the restroom and grab something to drink. So thank you for that. I think it is very, very elaborate. It's quite a lot. I see you prepared a lot of slides for us. So look, the good thing is that you've touched on the basis and where we could go with this. And this is going to be shared with everyone. So obviously a lot of people want a lot more information, including myself. as to weighing out the different options. There's things that you take into consideration. I mean, you obviously, if you're a startup, you're looking at budget constraints and volumes and that sort of thing. So each different method would require, or rather would have the pros and cons for different different size of organizations, as well as the different methods of separations, if you want to call them that. Yeah, I think that's really interesting. And then we're going to get that information, your slide. To start off with questions, ladies and gents, if I could ask you to raise your hands, I would love to hear what you have to say. We have a question coming from Pumza, or comment, Jeff. As you are having these products that seem to be so important with value of money, do you have a license or permit? If so, do you have your land to plow it, or are you getting primary product from someone else? If so, is there that person having a license for commercialization, or is he having a permit? We want to partner with people that are in the value chain of it after getting our permits, as we are having more land to plow but still suffering in getting a permit.
Yes, so perhaps I could speak to that. So we are, more in the equipment and the technical support about ensuring. So for instance, a good example is a lot of my clients are Adcock, Aspen, GSK. So those are the clients where we'll engage in terms of, a typical GMP manufacturing facility, come in to help with optimization and efficiency. So taking almost a gap analysis to the facility, making sure that, identify how do we get it right the first time? How do we make sure that you don't lose your money? I think it's really important for an investor as well to understand is, I might understand a lot about the cultivation, we've applied for a license or that license being approved, but I don't want to end up at the end of the day with product that doesn't leave because it's not meeting a certain standard. That I don't have a market where this can go. I'm quite flexible in terms of those approaches. There's a lot of products where we're in R&D. We try to operate, within the medical framework on licensing. I do work with a lot of universities as well to try and, almost get the right parameters set from the beginning so that we can almost take a tech transfer approach. Like I mentioned, Delta and Apex and Hydrocarbon, they've worked with so many different sites that, to scale the solution is relatively easy to do. It's one of, really making sure that it works and it works the first time. And that also we have local service and support, because I have many facilities where they have source materials or equipment and there's no support, no support around the qualification of that equipment, which is critical for the regulatory environment as it is. And there's no support often on what I do. Someone might buy an SPD system and they just don't know how to operate it safely as well, which is critical. So that's really where I try to drive my expertise. And the thing I would encourage stakeholders within government to consider is a central extraction facility, possibly for provinces. Key provinces where they can have a central extraction facility that's being run, that, biomass can be sent to from local individuals that have permits, licenses, or any other envisioned new improvement in terms of, collectives that want to send their product to a central facility. We have the technical capacity to support, and if there's a bigger role in terms of stakeholders, in terms of facilitating this, I'm really keen on that.
Sure. Well, I mean, we could obviously taking that offline and having a bit of a broader discussion surrounding that, because there are obviously some initiatives. Like I've worked very closely with Myrtle and Jules, the Dhaka couple, yes, regarding that. So that's an interesting conversation to have. Is Separations a South African company?
Yes, so just to clarify any confusion there, Delta Separations is an international ethanol extraction supplier. Separations is local. So we've got a Cape Town office and Johannesburg. I'm up in Johannesburg, but before COVID, I did a lot of traveling.
Where about in Johannesburg?
I'm in Randburg, so we're based in Randburg.
Where about in Johannesburg are you guys situated?
Randburg. So Randburg, and I'm in Fort
Okay, fantastic. Well, you and I have to have a conversation.
Yes, absolutely.
Okay, fantastic. Yeah, look, there's our audience currently today sits all over the country, so it's fantastic that you guys are in 2 major cities. I'm sure you'll be in touch. I'm liking the feedback. It says my connection is unstable, so I hope you guys can still hear me. The feedback's brilliant so far. We've got Stephen Kuno, who says big up. We've got Jackie Ramage in the comment there saying that separation supplies the equipment only. And to touch on that as well, you did say that training is a part of this as well. It's pointless just supplying the equipment and it's not being utilized properly. You're not going to get efficiency.
To that point, I mean, that's the most important. I often feel this is overlooked. Everyone wants a third quote and wants to look at the costing of the equipment. But the reason we've been successful and the reason we work with established universities and pharmaceutical companies and startups is you don't want your equipment down, not for a day. Very often you cannot afford, if you're a commercial operation, you cannot have your equipment down due to a lack of a spare part, a seal, something just silly, and you've bought it from China or you've bought it from someone you don't know in the US, and you sit in a situation where you're just not able to get the operational throughput from that equipment. That's intolerable. You know, a month or a week lost in terms of processing is almost as good as buying new equipment sometimes, depending on how you're doing it. So maybe a bit exaggerated a week, but I would say that it happens a lot. I see it all the time where customers reach out and say, I bought this, I can't qualify this, I cannot run this, I cannot support this. And the money's been burnt out of the pocket of the investor. And we try to avoid that from the beginning. We want to make sure that everything's going to run from day one, which makes us typically more expensive due to that expertise, but we don't repeat ourselves. We do it right the first time.
That's an important value proposition. There's tons of product on the market and anyone can find whatever it is they want online and in China. But without local support, you're going to be on the back foot. So I think that's brilliant. Another question from my side, you mentioned some of the names of the clients that you service. So it seems a lot like, big pharma is sort of the main market. Is that your core focus, or are you also, looking at smaller facilities, startups, and that kind of a thing as well? Is that a big part of your focus?
So I would say I actually get the best engagement sometimes from startups. So, individuals, organizations that, sometimes high value net individuals or collective groups that are looking to do this. They're the most forward thinking. I think it is realistic for pharma to take a bit of a cautious approach and a calculated approach to the industry, which is what we do as well. But we try to leverage that skill set to introducing that to our, let's say, more first-time adopters, someone who's getting into it for the first time, the understanding that QMS is not something you just buy. So you don't just buy regulatory documents and it works. You've got to work on those and you've got to refine those SOPs. You've got to make sure your site master files are right. You've got to almost have a compliance culture to that business. And if you do that from day one and you work in this constant approach of improvement, this Kaizen principle, constantly improving, you get to a business that if you do it right, you really can be unrivaled in terms of there's a lot of market share out there for a good extractor, good facility. If you get your extraction right, there's a lot to be done.
Fantastic. Okay, so we're just gearing up for our next speaker. I just want to share with you some of the comments. It's brilliant. Everyone seems to be enjoying it. I mean, we've got Ross McPhail saying, great presentation, Jeff, very informative. Thank you, we'll be in touch. Pumza, she commented earlier, Jeff, we need you in Eastern Cape. We are done and we are ready. Brilliant woman, her energy just exudes passion. So, you're going to really enjoy chatting with her. She's doing a really big operation down there, very hands-on, and there's a lot going on in the Eastern Cape. We obviously were there probably I think in Feb during the last conference there. So they're stirring up quite a storm. We've got Jackie Ramage who says, thank you, Jeff. As always, super informative and amazing knowledge. Roseanne, thank you, Jeff. Great work on the presentation. And Shana Jackson, very valuable presentation, Jeff. She's also got a question. I'd like you to answer this one offline just for timing, but you can have a look at it. It's in the chat. What does one look out for on the certificate of analysis to determine what the raw material is of good quality for various applications? I'm specifically interested in cosmetics. So maybe just delve into various applications and as well cosmetic. I think that's a very, very good question for everyone, but you could just do the response in the chat on that one. And then lastly, Sean, this was very informative, really very informative, Jeff. I'm definitely going to be in contact soon. Very interested in CO2 supercritical method for a commercial operation where we are in the process of setting up. Thank you very much. So yeah.
Excellent, Dave. Thank you for hosting this event. I've been enjoying it this whole morning. So I'll put my details in the chat as well for any of the individuals. And then if there's any questions, I'll just put 1 or 2 comments in there while the next speaker gets going. Thank you so much.
Fantastic. Perfect. We've got one more there from Nono Nokaya. Well done, Jeff. The Eastern Cape certainly needs your extraction center. So you're definitely getting some heat from the Eastern Cape.
Oh, I go to Aspen and Fresenius Kabi regularly. So I'll be down in the Eastern Cape hopefully very soon.
Perfect.
Excellent.
Thank you, Jeff.
Thank you.
We'll talk soon. Okay.
Okay, cheers. Bye.